Hi, hello. Let's talk about phylogeny. So, phyle means leafy branch, and geny means how something is developed or produced. So, it's a leafy branch of how things have developed and how they're related. Basically, a tree of life. So, there are some other fields of biology that do very similar things, but they are distinct. So, I want to distinguish them here. Phylogeny is a tree of how organisms came to be. It studies the evolutionary history of species and groups. Now, taxonomy, we have covered a little before. Do you remember that odd question I asked you? Do kangaroos prefer cake or frosting, generally speaking? That was that mnemonic device to remember the levels of the Linnaean hierarchy. Oof, y'all know my computer drawing is rough. So the hierarchy was domain, kingdom, phylum, class, order, family, genus, and species. Each level was called a taxon, and the last two taxa are used to name organisms like us, Homo sapiens, or kangaroos, where there's 4 extant species, and the red kangaroo is named Ostfronter rufus. Systematics is a broader field that classifies organisms and determines evolutionary relationships. So you can think of systematics as this overarching giant umbrella system that includes both phylogeny and taxonomy. This lecture is about phylogeny. Phylogenetic trees are branching diagrams that represent the evolutionary history or relationship of a group of organisms. The branching diagram represents hypotheses about evolutionary relationships. Remember that a hypothesis is a prediction. So scientists use observable physical characteristics like morphologies, the fossil record, and molecular data to predict how species are related. Phylogenetic trees can be represented by vertical trees or diagonal trees, but both of these trees are portraying the same relationships, and we will mostly look at vertical trees for this lecture. So here we're looking at a phylogenetic tree. A rooted tree includes a branch to represent the most recent common ancestor that connects all of the descendants. If this branch that represents the ancestor was not included in this tree, then this wouldn't be a rooted tree. A branch point, or a node, is where one line splits into 2 or more, where lineages diverge. So the 2 marker here represents a branch point or node, marker 3 and marker 4 represent branch points or nodes, and marker 5 is a branch point, but notice that it branches into 3 different taxa. Whenever you see more than 2 lineages branching off, that's a polytomy. The poly in polytomy means many, so many branches are forming because there's an unresolved pattern of divergence. We might not have enough information to distinguish how these 3 species diverge from one another. This taxon, taxon G, branches directly from the ancestral lineage. So you see the ancestral lineage at branch point 1, and then taxon G, where no other species have branched off. This is called the basal taxon. This lineage diverged from all other members early on in history. You might remember the basal taxon as being basic because it doesn't have as interesting and complicated of a history as the other taxa. And there are other types of taxa, like sister taxa. So sister taxa are groups that share an immediate common ancestor. So B and C are sister taxa, and D, E, and F are sister taxa. Rotating the branches of a tree around a branch point does not change what they convey about evolutionary relationships. That means that the order in which the taxa appear in this list at the branch tips is not significant. What really matters is the branching pattern. So between these 2 trees, the one on the left and the right, the tree is being rotated at the highlighted blue nodes, but it still tells the same relationships in the tree to the right. So humans and chimps are still sister taxa. Humans and chimps are sister taxa in the first tree, and humans are more related to lizards than they are to frogs. So humans are 2 branch points away from lizards. That's when they share a common ancestor. And humans are 3 branch points away from frogs. That's where they share a common ancestor. And in the second tree, where the nodes have been rotated, humans are still 1, 2 branch points away from the lizards, and humans are 1, 2, 3 branch points away from frogs. And fish is the basal taxon for both of these trees. When constructing phylogenetic trees, phylogeneticists try to construct the most parsimonious trees. Sounds like a fancy word, but it is not. Parsimony can mean cheap, so using as little money as possible. But in this case, scientists try to use as little explanation as possible to predict the most likely relationships. So let's say we were given the molecular data for a DNA sequence at a particular site in a gene for 3 seemingly related species and an ancestor. So the ancestor sequence is AGTT. The species 1 sequence is CTAT. Species 2 has CTTC, and species 3 has AGAC. So off bat, I would draw species 3 as the basal taxon because its sequence is most similar to the ancestral sequence, whereas species 1 is similar to the ancestral species only at one site, and the same is true for species 2. So this is how I would draw my phylogenetic tree. Now, if you don't have the same experience identifying these similarities and differences, you may have to draw a few phylogenetic trees to figure out which is most parsimonious. In this first hypothetical tree, let's count the number of events that would have to happen, meaning the number of changes in bases for this tree to be possible. So the basal taxon here is species 1. So when we compare the ancestral species to species 1, they are only similar at one site, site 4. So 3 sites had to change, 3 events had to happen for species 1 to diverge as the basal taxon. And that's why there are 3 tick marks on this lineage. Now let's take a look at how species 2 and 3 differ from the ancestral species. Remember that species 3 differs from the ancestor at only two sites, sites 3 and 4. It's possible that there was first a mutation at site 4, a change from T to C, which applies to both species 2 and 3. So the tick mark is made here, before the branching point for species 2 and 3. And from there, the sequence had to change one more time at site 3 to give rise to species 3, and it had to change 2 more times at site 1 and 2 to give rise to species 2. You would do the same exercise, counting the tick marks to count how many events are required for that hypothetical phylogenetic tree. When you count them up, this first tree has 7 tick marks, or 7 events. The second tree has 7 events. And this last tree has 6 events. So that means that this last tree would be the most parsimonious tree. Not because it has the least number of branches, but because it requires the least amount of events, the least amount of explanation. So what can we learn from phylogenetic trees? Trees show patterns of descent, not necessarily phenotypic similarity, not necessarily physical similarity. Although closely related species may look alike, it really just shows how species have diverged into different species. Taxa that are next to each other, that share a node, share a common ancestor. So for example, chimps and humans share this node, we share a common ancestor. However, please do not misinterpret. Humans did not evolve from chimps. Humans did not evolve from chimps. We simply share a common ancestor. 2 taxa are more related if they have a more recent common ancestor, and less related if they have a less recent common ancestor that's more branch points away. So humans are more closely related to chimps than we are to lizards. Humans are 1 branch point away from chimps, and humans are 2 branch points away from lizards. I could also say that chimps are more related to lizards, and if we trace back, chimps are 2 nodes away from lizards. So chimps are more related to lizards than chimps are related to fishes, because chimps are 4 nodes away from fishes. Trees do not indicate when in time a species evolved or how much change occurred in a lineage. So some trees can denote time and it will say it, but for the trees that we'll look at, the lengths of the lines do not mean that more time has passed, and the change that occurs at any branch point could be a single base pair or several hundreds of base pairs. To construct a phylogeny, systematists gather information about morphologies, like the structure and shapes of things, and biochemistry, like the different proteins and metabolites within their body, and the genes of living organisms. Phenotypic and genetic similarities due to shared ancestry are called homologies. Organisms with similarities in their body shape and structure or DNA sequences are more likely to be closely related than organisms with different structures and sequences. But we must be careful because not all similarities are homologies. Homologies are similarities due to shared ancestry, but an analogy, also called a homoplasy, is a similarity due to convergent evolution, not shared ancestry. And both morphological and molecular analogies can be confused for homologies. So how can we tell the difference between homologies and analogies when constructing a tree? You need to look beyond the superficial resemblances. If two features look alike, we need to really examine how similar they are based on underlying structure, how those structures are attached to other body parts, examine the fossil record, and ensure that similar levels of complexity exist in addition to looking at DNA sequences. The more elements that are similar between two complex features, the more likely that they are homologous. That's why the forelimbs of the human, dog, bird, and whale, pictured to the right, may not immediately look similar externally, but internally, the individual bones and the number of bones they're attached to are very similar. And there's molecular data to support that those are homologies. Now, similarities between the Australian mole and the African golden mole are analogies. Their similarities, they look fairly similar, but their similarities result from adaptation to similar lifestyles, to similar environments, but their species are evolving in two totally different locations, and so their similarities are not due to shared ancestry, and that is also supported by molecular data. Here's a new term, cladistics. Cladistics is the approach to grouping organisms by using common ancestry as the primary criterion. So if phylogeny is determining the evolutionary relationships, then cladistics is just talking about those groups. So a clade is a monophyletic group, meaning it is a group of descendants and the ancestral species that connect them. Monophyletic means one branch. So you could think of it as one branch of a tree and all of the descendants that diverge from that branch. But you have to include everybody, all of the descendants from that branch, because if you don't, then you could be talking about a paraphyletic group. Paraphyletic groups consist of an ancestral species and some, but not all of the descendants. Para can mean beside or abnormal, like paranormal activity is abnormal. Paraphyletic groups are not normal, like the all-inclusive monophyletic group. So in this group highlighted in blue on this phylogenetic tree, we're including the ancestral species that leads to these descendants, but we left out G. And maybe that's just because G was not pertinent to the conversation that we're having about this highlighted paraphyletic group. Another key group term is the polyphyletic group. Remember what poly means? Poly means many. So polyphyletic groups discuss many distantly related species, but it does not include their most recent common ancestor. So you can see in the phylogenetic tree highlighted here in blue, we see some distantly related species, and it does not include their shared common ancestor, which is actually this first branch point here, since we're including group D. Now let's put this into practice. In this phylogenetic tree, we can determine how ungulates, which are mammals with hooves, are related to hippos and cetaceans like whales and some other organisms. This blue highlighted area includes ungulates, hippos, and their common ancestor at this branch point. Is this an example of a monophyletic, paraphyletic, or polyphyletic group? I'll give you 5 seconds to think about it. Pause if you need to. It's a paraphyletic group because it includes the descendants and their ancestors, but it does not include all of the descendants. Without the cetaceans, this is a paraphyletic group. If cetaceans were included, then it would be a monophyletic group. How about this other highlighted area in yellow, which includes cetaceans and seals? Is this a monophyletic, paraphyletic, or polyphyletic group? I'll give you 5 seconds if you'd like to pause. This is a polyphyletic group because it includes more than one species that are distantly related, but it does not include their ancestors. So what else can we learn from phylogeny? Well, we can distinguish between ancestral characters and derived characters. A shared ancestral character is a character that originated in the ancestor of the taxon. A shared derived character is a new character unique to a particular clade. So for all of the organisms in this tree on the right, what do you think is an ancestral character? I'll give you 5 seconds. Having a dorsal nerve cord is an ancestral character that all of the descendants have. They all have a dorsal nerve cord, or else it would have been noted the loss of a dorsal nerve cord. What do you think would be an example of a derived character? Four walking legs is one derived character that did not exist in most ancestral species, but it is unique to this clade down here that includes frogs, turtles, and leopards. Know that losing features and gaining features can be derived characters. For example, snakes and whales evolved from organisms that once had 4 legs. Snakes and whales don't have legs, and that loss of limbs can be considered a derived shared character. And naming shared ancestral characters and derived characters is relative. It depends on who's included in the tree. If the tree you were given only considered the clade that I circled in red, then 4 walking legs would be considered the ancestral character. And the amnion, which is a membrane that covers the embryo during development, would be considered one of the derived characters because it only exists in these later diverged groups and not in the ancestors. Phylogeny can also be used to answer questions of homology and analogy. Based on this phylogenetic tree, I want to ask you, is the lack of limbs in snakes and eastern glass lizards homologous or analogous? I'll give you 5 seconds to think about it. Take a look at snakes and when the no limbs feature arose. Now take a look at the eastern glass lizard and when no limbs arose. According to this tree, the no limbs feature arose separately in these groups. And it wasn't an ancestral character that was passed to all of the groups. So having no limbs is an analogy between snakes and the eastern glass lizard. Let's try another question. CT are tiny microscopic hairs that some organisms use to grasp and stick onto things. Based on this phylogeny, is the lack of CT in iguanas and eastern glass lizards homologous or analogous? Let's look for the lack of CT on this tree. It doesn't directly say the lack of CT, but it looks like the presence of CT arose when the geckos split off from this common ancestor. This means that the common ancestor did not have CT, and all of these descendants also don't have CT, or else it would have been noted in the tree. That means that because iguanas and eastern glass lizards and their ancestor don't have CT, that is a shared ancestral character. We can use phylogeny to predict features of an ancestor that no longer exists from the features of its descendants that do exist, that are extant. For example, a 4-chambered heart is found in crocodilians and birds. There are other organisms related to crocodilians and birds in this clade, like the dinosaurs, but they're extinct. So I don't know much about their heart. The info that we do have suggests that the common ancestor of crocodilians and birds also had a 4-chambered heart. Okay, let's pause on the new info and review an earlier topic. I told you that phylogenetic trees can be rotated around a node or branch point and still tell the same relationships. So take a look at these 2 sets of trees, these 2 pairs of trees, and ask yourself, are these the same tree? So go ahead and work on that, pausing the video now, or else I'll give you the answer in 5 seconds. For question A, we see that the group W is the basal taxon in the first tree. And in the second tree, it's the same. If we look at the relationships more closely, tree 1 says that Z and Y share a common ancestor, and they are considered sister taxa. The second tree says that Z and Y are not sister taxa, so I immediately know that these are not the same tree. For question B, we again see that group W is the basal taxon. And in this first tree, Z and Y are sister taxa, and they also appear as sister taxa on the second tree. X and Y are separated by 2 branch points in this tree, and in the second tree, X and Y are again separated by 2 branch points. So these are the same tree, rotated around the second branch point. Nice job if you got it. So on this last phylogenetic tree, I'm showing you the tree for the 3 domains of life, BAE, or bacteria, archaea, and eukarya. This tree is based on similarities in DNA sequences that code for rRNA. So it's a little review from Bio 115. Remember that DNA is transcribed to RNA, and that's translated into proteins. Well, rRNA is ribosomal RNA, and ribosomes have that essential role. Do you remember their function? Ribosomes make proteins. So the DNA that encodes rRNA doesn't change very often. Or if it does change, it doesn't pass down those mutations often because a cell with defective ribosomes doesn't last for that long. So rRNA is pretty reliable for determining evolutionary relationships. And LUCA stands for the last universal common ancestor. It's the last ancestor that connects all life on Earth, represented by this first branch point. It was likely a single-celled prokaryote that lived in a hydrothermic vent, a very, very hot opening on the ocean floor. It didn't even know that it would give rise to literally every single living thing on the planet today, which is pretty cool to me. And next lecture, we'll talk about the major events that occurred before LUCA's existence, during LUCA's existence, and after LUCA's existence that gave rise to life as we know it today.